All right, so chapter 11 is focusing on chromosome structure and organelle DNA. Okay, chromosome structure and organelle DNA. So, as you have already seen, each chapter opens up with some type of case study or relevance to what the chapter is going to be about. This picture here that you're looking at is a picture of orphans from Romania. And scientists, or specifically geneticists, wanted to study a phenomenon that they observed dealing with hardships encountered early in life and how they can play a role in the shortening of telomeres. So before I explain a little bit about the study, I want to explain what telomeres are. Okay, your telomeres are found at the ends of your chromosomes, and they are what we call protective structures, and they're found specifically on the ends of linear chromosomes. But they also carry a record, right, in their length. So let's say if you experience a lot of stressors or issues that may cause impact you, it can be found on your chromosomes, the ends of your chromosomes. Evidence in previous studies have suggested that telomeres shorten with age, and these processes that you experience, if detrimental, can lead to the premature shortening of your telomeres, in addition to the factors that would not normally occur with aging. So let me expound on that for a second. And understanding your telomeres have every cell in your body, and in multicellular organisms has telomeres that indicate the lifespan of the cell. Okay, and as you age, you see this change in your telomeres. However, studies have now found, or what they call as new evidence, suggests that in addition to the aging that you would see of your telomeres, or the shortening due to aging, stressors or factors that you experience early in life can also impair the lifespan of your telomeres. Okay, now why is that important? That is associated with the lifespan of a cell. Okay, so back to this image or this diagram of these orphans. The scientists wanted to understand the impact of our early life experiences and how they may impact our telomeres, specifically our chromosomes. So to study the effects of early life experiences on telomere length, these geneticists studied 100 children that were living in a state-run orphanage in Russia. And at an early age, some of these children were placed in foster homes and others remained in the orphanage. Some studies that were further done demonstrated that the children in these orphanages received less individual attention and care compared to those that were fostered and grew up in a natural, more loving environment. Okay, so ultimately, institutional care was thought to be associated with more stress than foster care. When the children, now what's interesting about this study is that it followed these children for a period of time. So when these children were 6 to 10 years old, scientists went back and collected samples of their DNA and measured the length of their telomeres. And the results were actually very, very interesting. Okay. The results from the study suggested that the children that remained in the orphanage had significantly shorter telomeres than those who spent time in foster care. The researchers concluded that the telomere length is affected by childhood adversity. Children reared in stressful environments are more likely to have shorter telomeres than those raised in less stressful environments. Okay, and there have been other studies that have supported this because they looked at also abuse and chronic illness and how those things or factors affect your telomeres as well. But the importance is, and the take home from this case study or this opening portion, is that our DNA, our chromosomes, can be affected by trauma. Okay, and it leaves an impacting or footprint in our DNA. Okay, so this lays the foundation for what we're going to be talking about. So let's talk about DNA. Okay, and I want you to ask yourself a couple of questions as you're going through this chapter. What is the difference between DNA and chromosomes? Okay, so think about DNA. DNA is 100, and we're talking about specifically E. coli. It is 100 times longer than the cell that it is compacted within, that it is placed inside. So if you were to take DNA out of a cell, specifically an E. coli cell, and allow it to be linearized, okay, it's 100 times longer than the cell. So the question is, how is DNA that is 100 times larger than the cell itself able to fit in a small space like a cell, which is microns in size, so small that it cannot be seen with the naked eye? One of the factors that contributes to that is supercoiling. Okay, you have positive supercoiling and negative supercoiling. Okay, positive supercoiling and negative supercoiling. The enzyme that controls or regulates this supercoiling is an enzyme called topoisomerase. Okay, topoisomerase. So you can see here in this diagram that showing you DNA when it is supercoiled, either positive or negative. Okay. And how that affects the structure when it starts to twist and turn on itself. Okay. What I want you to do is, if you have a lanyard with you, I want you to take that lanyard and I want you to begin to twist the lanyard. Okay. And you're going to see and compare the size of the lanyard before you twist it, and then compare the size of the lanyard as you begin to twist it. And you're going to see that as you begin to twist the lanyard, the size or the length of the lanyard decreases or shortens because of the twisting. And this is what we see with our DNA. It helps with the condensing of the DNA. Okay. The the bacterial chromosome, remember, is circular. Most bacterial genomes are circular. We talked about that before, and they appear as clumps in the cell. Remember, a bacteria is a type of prokaryotic cell that does not have membrane-bound organelles. So the DNA is located, or the chromosome is located inside of the cytoplasm in a dense region called the nucleoid region. Okay, the nucleoid region. And this is what you see here. This is a picture of a prokaryotic cell where the membrane's been pierced and the chromosome has kind of spilled out. Okay, and you can see there's twisting and loops of that DNA. So it's highly folded or compacted. Now, eukaryotic cells is a little bit different. Okay, eukaryotic cells have a chromosomes that are linear, and their linear chromosomes can be further divided into different types of chromosomes, whether it's euchromatin or heterochromatin. But one of the important factors that helps with the condensing of these chromosomes in eukaryotic cells is what we call histone proteins. Okay, histone proteins. Furthermore, all chromosomes have what we call permanent heterochromatin. Another term for that is constitutive heterochromatin. And this would be synonymous with your centromeres and your telomeres. Okay, we can also see heterochromatin during certain developmental stages. Okay, this would be an example of facultative heterochromatin. An example of facultative heterochromatin is on one of the X chromosomes in females, when one of the X chromosomes becomes inactivated. So let's see. Okay, this table here helps to provide some characteristics of the difference between the two. So you have euchromatin, heterochromatin, right? Euchromatin is going to be less condensed. Heterochromatin is going to be more condensed. Where is it located? Euchromatin is located on the chromosome arms. So this is going to be the the areas of the chromosomes that have unique sequences that code for protein, that code for what are involved in gene expression. The heterochromatin is going to be more functional chromosomes. So it's going to be your centromere, your telomeres, and other places that tend to be more repeated sequences and don't code for protein. Okay. What you'll also see as you go through this table is when they're replicated. They're replicated at different times. Of course, it's in the S phase, but different times of the S phase. So let's go into a little bit more about these chromatin structure. Okay. So specifically, you have the nucleosome, okay? And a nucleosome is a eight figured or a component of eight subunits that make up a conglomerate of histone proteins that are involved in the condensing of chromosomes, okay? What are these components made up of? You have H2A, H2B, H3, and H4. The H stands for histone, and there's four histone proteins that appear twice. That's what makes it a what we call an octamer, okay? Because it's composed of 8 subunits of those 4 histone proteins that I just mentioned, and that forms the nucleosome. The nucleosome is a octamer type structure, okay? Pool, it's like a pool. Think of needle and thread, okay? The thread that's on a spool, okay, that allows the needle, the spool, the thread to be condensed and not, if you were to pull it off the spool, it would be very long. So you condense the thread by wrapping it around the spool, okay? Think of the nucleosome as that spool. And the DNA wraps around the nucleosome that allows it to become condensed. However, there are portions of the DNA that do not bind to or wrap around the nucleosome. The DNA that is not bound or wrapped around a nucleosome is called linker DNA. And linker DNA interacts with another protein called the H1 protein or histine 1 protein. Okay, histine 1 protein. There is what we call a higher order of chromatin structure, and this has to deal with how condensed the chromosomes are. And actually one is missing. So on this PowerPoint it says 30 nanometer fiber, but there's actually one that is 10 nanometer fiber, and this is synonymous with the beads on a string structure. Okay, and then you have the 300 nanometer, and then you have the 250 nanometer. As the numbers increase, it is synonymous with the condensing because it's measuring the width of the chromosomes, as you see here. Okay, so this is the simplest form of the chromatin. This is DNA as a double helix, and then here you have the nucleosome, this pink structure here. Notice it is composed of, it's an octamer, so it's composed of 4 proteins repeated twice, and notice how the DNA is wrapped around it. Okay, this yellow structure here that you see is the H1 protein. That is where linker DNA binds binds. And you can see here, this is like the 10 nanometer fiber. Okay, so it's not as condensed. This would be associated with what, chromatin. Okay, and as the DNA becomes more condensed, meaning it's wrapping around the nucleosomes tighter, it's engaged with the H1 protein, and then it also starts to form loops. It becomes more condensed, and the more condensed the chromosomes become, the increase in the width of the chromosome occurs, and this ultimately gets to a place where we have these discrete chromosome bodies. Okay, this is where we get chromosomes from. So this is chromatin, chromosomes. This is DNA. Okay, so hopefully now, while we're 10 slides in the lecture, you should be being able to develop in your own words, what is the difference between DNA, chromatin, and chromosomes? Okay, so here is your core histone nucleosome. There's your linker DNA. Okay, now what's important is that while the nucleosome is the fundamental unit of a repeating chromosome, nucleases, which are a type of enzyme that can degrade RNA, or nucleic acid I should say, allows for the potential nicking or breaking of the DNA that is not bound to the nucleosomes, also known as linker DNA. Okay, and you'll see here at the bottom, these are the 4 subunits that I was telling you about that are repeated twice that make up this octamer called the nucleosome. H2A, H2B, H3, and H4. All right, so let's go into the chromosome structure, okay, because the chromosome structure changes, right? You have what we call a polytene chromosome, and this is where you have repeated rounds of DNA replication with no cell division. So it forms like this puff, chromosomal puff, okay? And you have an enzyme called DNase 1, DNase 1 sensitivity, and this is where it correlates with what we call gene activity, okay? Gene activity. You see polytene chromosomes in Drosophila. and some other organisms, but in specifically in Drosophila. Okay, and this again is associated with instances where you have repeated rounds of replication, but you don't have cell division. So the DNA keeps replicating and duplicating, but it's not dividing into daughter cells, and so you have this large amount of just repeated chromosomes. And that's what you see here. It's chromosome puff. You see that here as well. All right. So I mentioned DNase 1 activity. I want to show just briefly, I won't stay on this, but DNase 1 sensitivity is correlated with transcription of some globin genes in chick embryos. Specifically the U gene, it encodes embryonic hemoglobin. Okay. And there are different subsets of these genes, but you have the D and the alpha genes that encode the adult hemoglobin. And the red arrows that you see here in this diagram show sites or indicate sites where DNase 1 can digest the DNA. Okay. So this diagram or this image here that you're looking at is showing methylation. Now, I previously mentioned in a previous chapter about methylation, and this is where you add methyl groups, that CH3 group, to DNA, and it can alter gene expression. This is what we see here. Specifically, you see this variation in the coat color of these different mice, and it's due to a gene called the agouti gene. Okay, the agouti gene. And methylation, which in the DNA, which causes chromatin structural changes, can alter or induce what we call epigenetic changes in genes that will alter the coat color or the pigmentation of the coat color in these various mice. So let's look at the centromere. Okay, the centromere again is one of the types of heterochromatin that is essential in many aspects and processes of the cell, specifically dealing with cellular division. Okay, the centromere is this restricted or constricted region of the chromosome where those spindle fibers attach. And what's important is it is repeated DNA. So it doesn't code for protein, but it's important because when a chromosome lacks a centromere, it can be lost in mitosis. And you'll see as we get further into the semester that this is in part thought to lead to some instances of Down syndrome. Okay, so having the centromere, okay, that you see here, that's the centromere, is important for cellular division and many processes of the cell. The telomere is essential because the telomere, again, as we talked about before, is essential for providing the age, the lifespan of a cell. Okay, these centromeres were first discovered in 2009. Well, they were given, the individuals that discovered them were given the Nobel Prize in 2009. It was Blackburn, Greider, and Salzak. And they were awarded the Nobel Prize in Physiology and Medicine for discovering the structure of telomeres and how they replicated. So understanding that telomeres also provide the lifespan of cellular aging, but they also provide structural integrity of the chromosomes in a form of protection. Okay, this is just showing you the sequences that are found in telomeres of some organisms. And so here you can visually see the DNA sequence of the telomeres. And this is this portion here that is no longer double stranded will actually form that T loop and allow it to fold back on itself. And this helps to what we call stabilize the ends of the chromosomes. So the chromosome components or the chromosome for each cell type is different, specifically with a eukaryotic cell. Okay, there's various what we call genetic variation or sequence variation. Okay, this refers to what we call the C value. Well, the C value is the different amounts of DNA per cell. Some have thought that there was a direct correlation between the complexity of a organism and its C value, but we're finding, as you'll go into more in depth of this in molecular, that is not always the case. That's why they refer to the C-value paradox, which you'll learn more about. Okay. But just really quick, to go into this, not too much yet, but denaturation. Okay. Denaturation refers to the ability to denature, to disturb or disrupt the bonds that hold together DNA molecule. Well, we know that two strands of DNA are held together via hydrogen bonds and, of course, other electrostatic forces, but primarily hydrogen bonds. Okay, so the rate at which DNA can be denatured is the melting temperature, and this varies based off of the genetic composition of the DNA. What do I mean? Go back to when we talked about the DNA, specifically the bases. Guanine and cytosine are held together by 3 hydrogen bonds, adenine and thymine, 2 hydrogen bonds. So if you have a molecule of DNA that is heavier or more concentrated with cytosine and guanine, the melting temperature is going to be higher than another molecule of DNA that has less cytosine and guanine due to the cytosine-guanine ratio. Renaturation refers to the reforming of those hydrogen bonds between those two bases, allowing the two strands, to reform that hydrogen bond and reanneal, what we call reanneal together. There are different types of sequences in DNA, and oftentimes this may also alter the melting temperature, right? You can have unique sequences in DNA, and this is often can be associated with DNA. It can change based off of the gene family. So you have proteins that have similar sequences that are evolutionary connected, but these are usually sequences. Unique sequence DNA is DNA that is coding for protein, so it is unique. Okay, but again, there are some proteins that share similar sequences. They're not identical, but they're very similar, and so we group them as what we call gene families, and this is thought to be due to evolutionary changes and duplication of certain genes. A great example of that is the globin genes. Right, so this is just showing you the genome sizes, right? Look at humans. This is our genome size in terms of base pairs. One would think that one's genome size is associated with the complexity of the organism, but as we see here, corn has a higher genome size or greater genome size than that of humans. So this also again goes back to that C-value paradox, where the genome size does not always is not always associated with the complexity of an organism. All right. Some types of DNA sequences in eukaryotic cells. We talked about the unique sequences, which are associated with more protein-based or coding sequences. But then you have repetitive DNA. Repetitive DNA is associated with like your telomeres, your centromeres. This is portions of the chromosomes that have a more functional role. They don't necessarily signal for or code for protein, but they play a more structural or functional role. Okay. Then you have your moderately repetitive DNA. This would be like your tandemly repeats or your interspersed sequences, like your signs and lines, which are associated with transposons. And then you have your highly repetitive DNA. It's usually less than 10 base pairs long, and it's associated with what we call microsatellite DNA. Now, I want you to keep in mind that not, and especially in a multicellular organism, or a multi-organelle, like a eukaryotic cell, there are multiple organelles in a eukaryotic cell, and the genome that they possess, those organelles, like the mitochondria, the chloroplast, have their own DNA, their own genome that is separate from the genome in the nucleus. Okay, so in the eukaryotic cell, animal or plant has a genome in the nucleus, but then it has mitochondria, it has chloroplast, it also has its own genome, and what the genome is coding for is like proteins, of course, but ribosomal RNA, tRNA. Okay. Now, the etiology of the mitochondria, for example, is thought to be through what we call symbiosis. Okay, and this is where mitochondria and chloroplasts were thought to be once free-living bacteria, and they were engulfed by primitive eukaryotic cells, and now they live in a symbiotic relationship, meaning it's the mitochondria, as you can, it's very clear, benefits the eukaryotic cell in that it helps to provide energy, ATP production. Okay. So I won't, we won't go into too much on this, as I've already mentioned it, but this is just showing you how the journey of the symbiotic relationship or endosymbiotic theory that proposed the primitive relationship between these primitive eukaryotic cells and mitochondria and chloroplasts, which were thought to arise or arose from bacteria. All right. So these organelles have a major factor. So, and you all know, everyone says the mitochondria is the powerhouse of the cell. Yes, but the mitochondria also gives some information about inheritance. For an example, we call it uniparental inheritance, right? And this refers to the inheritance of mitochondria, which is from it's maternal, okay? If it's an animal, okay, the mitochondria is derived from the mother, the female parent, not the male. And so it allows you to see the inheritance and trace lineage through the mitochondrial DNA. This is actually a booming field in genetics, where they're able to do ancestral DNA and analysis. And then you have replicative segregation. And this is where you have each cell that has hundreds to thousands of organelles. And as replication occurs and division, these organelles are also dividing and replicating along with the same process. And so oftentimes you can have what we call heteroplasmic cells. And this is organelles that segregate randomly. Okay, and you can see this from this picture here, where you have a protist that is stained so that you can see the nucleus in red, the mitochondria is in green, and the mitochondrial DNA is in yellow. Okay, and this is showing you here the heteroplasmic behavior or heteroplasmic cell due to the various organelles within it and how they segregate. And you can have instances where you have most of the cells that have divided and produced offspring are heteroplasmic, right? But there are some instances where the cells will only receive one type of organelle, or they can, whether it's all mutant or all normal, as you see here. Homoplastic, meaning the same. Hetero, meaning normal and mutant, or all mutant, all the same. So what are some traits that are associated with mitochondrial DNA? You have petite mutations in yeast, okay? And this sometimes deals with, a lot of the mitochondrial diseases are dealing with aerobic respiration or some type of metabolic mutation. You have MRF, which is characterized by muscle twitches, weakness, i.e. myopathy, and progressive stiffness. And this is when the muscle cells of an individual that is affected, they can be viewed under a microscope, and they're very abnormal. They're very rigid. LHON, or Leber hereditary optic myopathy, is where you have a mutation in mitochondrial genes that encode electron transport proteins. And typically what is seen with individuals with this type of disease is that they have a sudden loss of vision in their middle age, as well as muscle weakness as well. So there is a direct correlation between humans, animals, and mitochondrial disease, mutations in the DNA that cause diseases in humans. And this is the example of the petite mutations that are deletions in the mitochondrial DNA. The mitochondrial genome is, they're much smaller than that of the nucleic acid in the nucleus, okay? And their sizes vary based off of its origin, right? Whether it is human mitochondrial or yeast mitochondrial or even from plants, but their size will differ. And this is another table showing you the in base pairs, the variation in the sizes of the mitochondrial DNA based off of the species. Again, this is why we can't always assume that the higher number of base pairs or genome size is associated with complexity. All right, let's look at the human mitochondrial genome. Okay, so in here you have the mitochondrial DNA. Notice it is circular. The circle here, okay, the outer circle represents the heavy strand, and the inner circle represents the light strand. You'll see the origin of where they start is classified or as the lowercase ori and then H or ORIL. And there are a host of genes that encode various enzymes and proteins that are needed to carry out certain functions, like NADH dehydrogenase. Okay, and this figure in B is just an electron micrograph of mitochondrial DNA that has been isolated. This is a diagram showing a mitochondrial genome of yeast. And again, you have the two strands, and you'll see that there are different genes that are present in this mitochondrial DNA, and also the variation of different tRNA genes, proteins, and ribosomal RNA. Okay, so the evolution that we see of mitochondrial DNA, one, there's a high mutation rate in vertebrate mitochondrial DNA, and a number of these genes in an organization remain relatively constant. Okay, relatively constant. Most of the copies of the mitochondrial DNA are identical. So what happens when there is damage, okay, to the mitochondrial DNA? There is a direct correlation between damage that occurs to mitochondrial DNA, whether it's genetic mutations, oxidative stress, and aging, okay? Great correlation between it, okay? One, as one ages, oxidative phosphorylation capacity declines. And so you start to see a decrease in that capacity. Also, there are many human diseases that appear suddenly and middle age to later on in life. And this has to do with, again, mitochondrial DNA. Okay. In chloroplasts, there are instances with chloroplasts that also experience that produce traits, but they're going to be, of course, in plants, right? And this is associated with cytoplasmic inheritance of chloroplasts having their own DNA and therefore experiencing issues with that or symptoms with that as well. And this is a diagram of the mitochondria DNA for rice.